All right, I think we're all energized completely with that last session. And our next speaker also has a lot to give you in terms of nuggets and knowledge. So, this is the second time I'm opening for Eric in this conference, and I would do this 1,000 times over because he always brings something new to the table. For those of us who have joined today, Eric is the director within the Statistical Innovation Center at Eli Lilly and Company and known for his our podcast and our weekly projects. He is so well known in this community that while he was joining, people were actually talking about the celebrity coming on. He's so well known that anything I say will fall short here. So, all I'll tell you about is that Eric will bring us the nitty gritty of successfully submitting an application to the FDA in his talk today. Keep your eyes peeled and your ears open. Here comes a talk full of knowledge and insights.
Well, thank you so much. It's hard. I admit it's hard to follow what Jacqueline just did, but I'm going to do my best. But I am really excited to share what's really been a culmination of a very long journey with many, many moving parts. But I think it is a great story to tell. So that is where I'm going to take you through the journey of what we now know to be the first successful, at least in the pilot phase, Shiny app submission to a health regulator like the FDA. So this talk is going to be a mix of everything, like most of the things I do, mix of technical bits and a bit of story time and hopefully some inspiration. But I must say that I am just one small part of this overall effort. You'll hear more about that towards the end, but in any event, I feel very privileged to be speaking to all of you with this audience on such a big effort. So I'm going to put my spin on where I think a lot of this started. And again, this is Eric's spin here. I think the seeds were planted by a very monumental event for many of us who are enthusiasts of open source using R in life sciences. And that was the first edition of the R Pharma Conference. We actually held this in person for the first couple of years over at Harvard University in Boston. It was a small group, but the fact that this was my first experience really meeting those at other companies that shared my values, but also, frankly, were absolutely brilliant with the ideas that they were proposing and the technology behind it, but also the way that we could change this industry is one of the most valuable experiences I've ever had. In fact, I honestly get a little emotional thinking about it because I think it's changed a lot of my you might say my work life, or frankly my inspiration to hopefully inspire others too. Now there are a couple pieces here that I want to highlight from this inaugural event that proved to be instrumental to where we are today. We had a keynote from the esteemed Joe Chang himself on responsible use of Shiny in pharma, which now in 2023 we're seeing there's a lot of it. So we're going to get to that later. But it was very interesting to see that this set the tone for the majority of the talks in that first year were about Shiny. That was not planned. That was all of us trying to push the envelope a little bit here and there. But boy, did I learn a lot in that first year. But I also want to highlight this was not just about industry peers in my industry here. We were also fortunate to have representatives from FDA at this very event. Where you see pictured in the corner there is Paul Schutte, who is one of their computing directors over there. And he enlightened us on many interesting ways that the FDA is evaluating novel open source technology. Now let's get to some of the things that the other things I want to mention is that, yes, it was great for all of us to get together at the conference, but what's happened after the conference is just as monumental. In my humble opinion, our pharma was the start of what eventually led to what we now call the pharmaverse. You heard this a little bit from Philip's excellent keynote earlier in the conference, but this is a huge shift into how those of us in life sciences are not just pushing the envelope technically with open source, but we're actually sharing it with each other in open source. This has proven to be a foundation that I think is going to transform this industry. And I don't throw that term lightly, but I am seeing the fruits of it already. And what we're going to talk about today is certainly has a lot to thank for these kind of efforts. And one of those that you actually heard yesterday was the shiny framework called Teal. Teal, if you missed that presentation, is part of the Pharmaverse as a way to quickly explore and visualize and summarize data sets, most likely clinical data sets. And the way that it's got the architecture behind it is absolutely amazing in terms of engineering, but also I think in the impact that it's going to have for clinical teams to interpret their data effectively, giving non-statisticians the power to review their data efficiently. This has been such long overdue, in my opinion, but Teal, I think, is going to set the stage for some amazing innovations in this space. The other nugget that we found out in these collaborations is that, yes, our colleagues at FDA are also using Shiny quite a bit. I have a few examples here, but the takeaway is much like how those of us on the industry side are using Shiny to transform our workflows, FDA is also using it to help with their particular workflows and to analyze novel uses of data. And in fact, I've had a taste of this as well from a mix of both. Well, let's dive into a little more, shall we? As I've said, Shiny is transforming these clinical workflows because what I've seen is that it's accelerating the speed that we can get to big internal decisions. As I mentioned with Teal as an example, we're finding much more efficient ways to review clinical data. And then also a part that I've been really concentrating on the past few years, putting an intuitive interface in front of what can be an extremely complex pipeline of analysis. My realm is a lot to do with clinical trial simulation, as well as novel uses of multiplicity control, which are not for the faint of heart, and it can be very difficult to program on its own. But the fact that we have Shiny apps in front of these complicated processes is making sure that many in our organization can take advantage of these novel uses of methodologies and again get to decisions quickly. And my taste in this from a external perspective is that I was able to be a part of what's called the Complex Innovative Designs Initiative that was funded by FDA a few years ago to invite company sponsors to push the envelope with how we're designing clinical trials. And so I was fortunate that my company participated in this, and we actually shared a Shiny app as a convenient front end to simulate clinical simulation results and to summarize them effectively. That was my first taste of ever sending something out of the door, so to speak, from a company over to regulators that use something like Shiny. Now, of course, with that in mind, let's think about our day-to-day, so to speak, as clinical statisticians and data scientists. In the end, we're likely submitting a treatment for hopefully approval to help patients. So if we're seeing Shiny transform a lot of these workflows here, how could it help a clinical submission package? How can we get away from just sending voluminous outputs? In the old days, it was in paper, sometimes in truckloads of paper, and now it's electronic form. But how can we make the use of Shiny transform the way that we're sharing these results? Well, this is a journey that has now been spearheaded by a very novel working group under the R Consortium called the R Submissions Working Group. You've heard about this a bit before, perhaps, in the conference, but this is, again, a cross-industry working group with members from both the life sciences side and regulatory agencies. We're going to keep hitting that point home as we go here. And the idea is that the group is going to conduct pilots on how clinical submission packages that leverage R and other open source for analysis programs and results and the feasibility of sending that to regulators. And of course, going through these pilots, our goal is to identify what are the gaps, what are the things that are missing in this process, what can we do better, what can we learn from, so that we can provide a reference, a starting point for sponsors to use as inspiration on their particular journeys for their next submission. But the key is this is all open. None of this is being done behind closed doors. This isn't some hypothetical situation. This is all available on GitHub. Every step of the way, all the development and even the collaborations, which I'll get to next in a little bit. This is all right here available. Again, from when I started in this industry to now, that's a big shift. And boy, I am here for it. Now, to get to the foundation, so to speak, the first pilot that was conducted in 2021 looked at the capability of using R exclusively to generate the summary tables and visualizations of what we would consider the tables, listings, and graph delivery, or TLGs, and how that could be transferred effectively to the FDA. And this is where we set up a repo and we set up collaborations with FDA, and we were able to submit a full self-contained mock submission, again in this pilot one. But the key point is that this adhered to what is the current mechanism of sending information to the FDA, which is called the Electronic Common Technical Document Specification. That's a mouthful. Let me tell you what that means in a nutshell. Basically speaking, all the R programs that we put as part of this submission package, and it literally is an R package that we made for this, has to be bundled as a text file. I mean, literally a text file, .txt, and then somehow unbundled back into the normal R structure so that the reviewers can actually use it. That was made possible by another open source contribution in this space called Package Lite, which, again, you'll get my opinions on this in a little bit about why this has to exist, but we have to keep in mind it's one thing to be aspirational, but we also have to be realistic as well. Right now, this is the reality of how we're sending submissions to regulators like FDA. So the fact that we can use R to help convert and back convert in this process is just opening more possibilities. So in this package that we sent, it was complete with the R code, again, bundled as a literal R package, along with the Atom data sets and the associated outputs. And it was submitted successfully in November of 2021. Now, you're asking, Eric, that's the static stuff. You said you did some shiny stuff, right? Of course we did. Guess what, folks? We're going to live dangerously. We're going to do the demo here. We'll see if things crash or not. And I almost crashed my system. No, I didn't. Not to scare you. All right. So what you're going to see here, let me see if I can make this full screen, is the pilot application that we have put as part of pilot 2 that is the meat of this talk today. This application, I admit, is not going to win any design awards, but this is absolutely massive in terms of what we're about to show here. So we have a little welcome page that kind of gives an overview of what is inside the application. But we're not going to read this verbatim. Let's kind of take a look at what's in here, shall we? So up front, what we have is a table of demographic summary. So if you're from this industry, this is typically always in a clinical submission, a summary table of your patient characteristics. Behind the scenes, we'll talk about the packages it's using for this table in a little bit. But again, this is the first output that was part of that pilot submission. Next, we got the, might say, the primary analysis table, which is looking at the efficacy variable of interest with some footnotes. Again, it's a table. What can you really say about it? But we also have an efficacy table for another endpoint analysis, in this case, a summary analysis on glucose. But we put some niceties in here, like little tooltips. Maybe it's just giving a little helping hand to what kind of participants were involved in this comparison. Also, what is this inference about? So again, you're starting to see Shiny give a little bit of help to this static summary, little bits and pieces, right? And then this table of how many patients, percentage of patients complete each visit. This was actually not part of the original Pilot 1 submission. But when our reviewers got a hand on the early version of this app, it kind of opened some possibilities to them of some additional summaries that would be helpful to have in one place for them to easily compare with the rest of the summaries. So we said, why not? Let's put that in. We'll touch on the iterative nature of this process in a little bit, but most of the time for Shiny, we want to get to some visualizations, right? So what we have here on the screen is what we call a Kaplan-Meier curve. You've seen bits of this, I believe, in the teal demonstration, where we're looking at the time to the first dermatologic event. We'll touch on this caveat in a little bit, but if you remember the presentation from yesterday about teal, what we have here are filters so that the reviewer can interactively explore what might happen if they do some, you might say, subsetting of the data here. So here, thanks to the power of the teal modules, I can now do a filter based on patient age from the analysis subject level ATOM data set. And as you see, the plot's changing on the fly. Numbers are changing below. Very responsive. Excellent, excellent responsiveness, as I said. We could add more if we wanted to, but it's all right here, interactive. And now you're seeing the power of Shiny right here. This is, again, something that all of us know on this conference audience, but now we're putting this in the context of clinical results that are going into a submission. So the app itself, very, you might say, utilitarian, but remember, the purpose of this pilot is to see if this is technically possible and what are the issues behind it. So let's talk a little bit about, let's say, go back to the slides here. Wrong tab. This is it. All right, let's get back here. Let's talk a bit about the technical stack behind this, because we all love our technical bits in this conference. So a running theme in this conference has been the effective use of Shiny modules. Well, you'll be happy to say that when I'm involved in an app, it's going to have modules no matter what. So this does have modules created inside for each of those tabs that you saw in the interface. And much like how Pilot 1, we actually assembled that as a full R package. We also used an R package scaffolding, the Golem framework, to turn this app into an R package. Now, whether Golem becomes a quote unquote standard for the future, that's to be determined, right? But it may not be a one size fits all approach. But in the context of this pilot, it seemed like a natural fit to extend our principles from Pilot 1 into Pilot 2 by wrapping the application as a package. But as a must, whenever you do any Shiny app development and you're sharing it with external collaborators or even internal collaborators, you must use RM or some kind of package management dependency system, because in this case, for us, we had to ensure that the reviewers could replicate the R environment that was used to build the application from an R perspective. Now, again, much like how I shared at the beginning here, everything we're doing is open source. So all the code behind this app is on GitHub. There's going to be links throughout this presentation that you can look at after this. So you'll see every step of the way, all the bugs we fixed, all the decisions we made, and everything is there. And then on top of sharing the app, much like in Pilot 1, we wanted to share what's called an analysis data results guide, so a reviewer's guide, so that it's easy for the reviewer to get up to speed with kind of the backstory of the files inside the submission package. But this also had a very important set of instructions on how the reviewers can run the application on their system. Now, keep in mind, this system that I'm talking about, at the time we made this submission, they don't have a standard, say, POSIT Connect server that they could deploy this with. They have to run this app locally on their machines. So this ADRG needed to have precise instructions on how they could take the submission that was sent in the ECTD gateway, unbundle it, prepare their R environment, unpack the package dependencies with RM to restore the package library, and then eventually run the application. Well, it did take a lot of effort to write that doc, but we had a little fun along the way, because guess what? We used Quartal to do it. And that actually paid us dividends as well, because we were able to get both a friendly HTML version of the document and a PDF version, really without much code changing at all in the document. So I love to use novel technologies in this space. Now, I'll take a little digression here because I have to give some quick plugs to the fact that I knew that the reviewers were going to be targeting a Windows environment for their setup. Now, some who know me know I actually don't run Windows anymore on my servers. I'm a Linux person all the way right now. So how was I going to effectively test and mimic as much of their environment as possible? So it's a little beyond the scope for today, but I want to give a huge thank you to Martin Wimpress, who is one of the leading Linux, you might say, distribution authors out there. He has created a project called Quick EMU, which is basically a very efficient way to spin up virtual machines on Linux hosts. And I use this immensely as I got the app ready for sharing, and I had to troubleshoot all the different possibilities of mishaps or other technical bits that could occur once they get the ECTD package downloaded on their system. So there are a lot of snapshots made, a lot of trial and error, throwing R off, throwing R back on. And I admit going in Windows is not my cup of tea usually, but hey, for this project, it was hugely important. So thanks to Quick EMU for making that seamless for us. Now, the other part I want to highlight here is that this was a very collaborative process. The fact that we're able to get regular opportunities to elicit feedback from the FDA was absolutely massive to make this successful. And in fact, the link in the slide there is from one of our working group meetings where a lot of these issues were hashed out, where we had members like Paul Schutte from FDA join us so that we could talk about these issues, talk about fundamental concepts. And plus, we were able to share the app externally on Shiny Apps.io as we were going so that they could quickly kind of see what we were up to without them having to download everything themselves right away, which again, streamlined the review process. It was really exciting to use that piece of technology as well. But another theme that we've had in this conference is that Shiny gives us tremendous power and flexibility when we're visualizing these results or interacting with these results. Well, in the context of a clinical submission, there's a few points we have to keep in mind, is that most of the time we have what's called a pre-specified analysis plan. They call it the statistical analysis plan, or SAP. That outlines verbatim the types of statistical modeling and inferences that we're going to use, potentially with subgroups of patients that are predefined. But as you saw in the app, we only had the filtering capability in the Kaplan-Meier plot, the visualization part. There was initial feedback when we first put that feature in. We had it for all the modules, but that could lead to potential confusion for a reviewer on the regulator side, that if we're allowing the user to do that filtering based on, say, age or gender or what have you, if we haven't pre-specified that in the analysis strategy, that could lead to what's called an unplanned inference. And so we made a compromise that, okay, well, the fact that the Kaplan-Meier plot didn't have the p-value associated with a model on that particular display, it seemed okay to at least showcase the power of Shiny in that context, but we ended up having to take it out of the summary tables. But this issue, I think, is not done yet. But the more importantly, this pilot was the way that we could talk about this together in the same venue so that we could think about the future and the power of Shiny and how it's used in this particular situation of clinical submission. So the story is not closed on that capability yet, but at least for this pilot, we got a great lens into some of the issues that all of us are going to have to think about as this becomes mainstream. But I'm happy to say that even with all this, that in in November of this past year, 2022, we had a successful transfer of the app to the FDA. So that deserves some celebration. Let's do one more here, because that was a monumental task that now we have a reference of a Shiny app that has been submitted to the FDA. Yes, I know it's a pilot, but in the end, up until this point, many of us are looking at each other, so to speak, and asking, have you submitted a Shiny app yet? Have you submitted a Shiny app yet? Well, guess what? At least in a pilot situation, somebody has, and it just happens to be a group of us working in the open. I think that's pretty neat, if I do say so myself. Now, this is not the end of this journey, so to speak. There are lots of new innovations coming, hopefully via new pilots in this R Consortium working group. I'll give a little snapshot here. We've got a pilot ongoing to create Atom data sets directly with R as part of the submission package. One that I will be heavily involved with this year is seeing how we can leverage state-of-the-art container technology to share a Shiny app. Now, as we know, we've heard Docker mentioned a few times. We're going to look at different technologies and see if that can streamline this distribution process. But we invite any of you that want to get involved. We have the website for the submissions working group, which, by the way, is also built in Quartal because Quartal was fun. We have links where you can get involved, links to each of the pilots, because this collaboration has been a huge part of this successful journey. Of course, I do not want to, I have to make sure that we acknowledge all the many brilliant colleagues that we've had in this working group across multiple pharmas and from health authorities that have been so instrumental in getting us to this stage. We have all benefited so much from each other. Again, I am just one small part of this, but the collaboration has been something I've been so excited to be a part of. And the fact that I can share my love of Shiny, love of R in a way that can hopefully transform this industry, like I said, I'm here for it and I'm here to stay for it. So again, that was a whirlwind, but I hope that was interesting to you. You can find me, maybe my voice a little bit on various channels here that I've linked to, but I certainly thank you for your attention. I thank you for watching, and I hope you're inspired to get into this and get into innovations in this industry like I have been. So thank you very much.
Fantastic. Thank you so much, Eric, and congratulations for completing that milestone. Such a huge ordeal. This is amazing. And we're all looking forward to, where this goes next. We have a few questions from the chat. So we have a question from Sivu, who says, where do folks typically store this data before it gets to Shiny? This is in reference to the dermatologic event KM plot, which you showed just now. And basically, the person has asked that they're trying to imagine the whole stack.
Yeah, excellent point. So when you get to the link to these slides afterwards, we'll have links to the GitHub repos where we put on GitHub the actual submission package itself. And in that submission package, you will see what are called transport files of these 4 or 5 different data sets that the Shiny app and then the pilot one, the R scripts, would simply source in to conduct the analysis to produce that figure. So those data sets are typically submitted as part of that actual package. So then the sponsor can, or the regulator can easily rerun those analyses for themselves and see exactly what data was the source there.
Great. So I think that answers the question. I think that would also add to the reproducibility part, which brings me to one other question about reproducibility. Does the FDA accept Dockerized apps? And this question was asked by RWeekly. And by what pilot we can expect Dockerized apps and submissions?
Yeah, so at this point, I know there have been attempts at this in other working groups in the past to look at a Docker container as a companion to a submission package. And admittedly, there were some hurdles. In fact, the biggest hurdle now is ironically Docker itself. So what we're going to address in this pilot that we have coming up, it's unofficially pilot 4, is that we're going to look at a technology such as Podman that still has like a compatibility with Docker, but is again an open source solution in terms of running it, to see if it can be feasible to put the application into a Docker container, because that would be way more efficient to share than the, you might say, the magic of bundling and unbundling through the ECTD gateway. But this is really pushing the envelope. There are a lot of moving parts there. So the story will be determined if this will be successful, but we're going to give it a go this year and see if we can find out.
Yeah, I think it's all about trying and finding it out. So I think that definitely answers that question. Now there's a question about pilot 2, which says it's by Dongsun Kao. Does pilot 2 include define.xml? Any progress on using R to create the define.xml?
So the scope of this particular pilot was not so much to use R to produce the define.xml. That was just kind of like a part of what we call the source that we were using based on some open CDISC data sets that we used as a pilot. But that is certainly something that I think will be under consideration, especially as the pilot of using R to generate atom data sets gets underway, of just how far can we trace back that stack of using R to produce these other submission deliverables. So I'd imagine there will be more to that story later on. This pilot, we wanted to make sure the scope was just about Shiny, just about the R produced results, so that we didn't admittedly muddy the waters a little bit of trying to do too much at once. So these have been strategically designed to kind of build upon each other a bit.
Fantastic. I think that definitely answers it, and I think that's the standard and great approach to do that. Now we have another set of questions, actually two questions that relate to each other, and they're less technical and more about uh, the process overall. So we have a question from chat from Damian. What was the biggest challenge you faced throughout the process from the formalities perspective, non-tech, and what was it like to collaborate across companies that otherwise are quite closed off due to IP and all sorts of protections in place?
Yeah, great questions, Damien. I think honestly, the biggest challenge is kind of like the unknown of this is that, as I said, no one on record has mentioned that they've submitted a Shiny app as part of a real clinical submission. So we're kind of, I don't want to say going this in the dark, but this is a new journey for many of us. So a lot of it, honestly, the biggest challenge is just how much flexibility do we offer in this app? And as I said, we started with a lot of flexibility, but then we found out through the iterative process that wasn't the best idea. And now it does make sense on a kind of practical standpoint. But I think the challenge will be just harnessing the power with responsible use of it, if that makes sense. So I think that's a evolving issue, but certainly with respect to the collaboration, this was easily one of my most, you might say, favorite efforts in order of making sure that I'm learning a lot from my industry peers, but also getting more real-time assessments from our regulator colleagues. It's rare that it's this real time, that it's been in the every month we will get together and hash these ideas out. I think that approach is something that I hope the rest of our collaborations can learn from, and it's a big reason why this was successful here.
Brilliant. I think you've paved the way to actually get this done, and I think with that answer, we kind of see that you kind of spearheading this entire thing. So this is amazing, and I think that's all the questions that we have right now. Thank you so much, Eric, for your time and for this amazing presentation, and congratulations once again for this amazing achievement.
Thank you for having me, and it's been an amazing conference. Again, thank you.